Okay, before we begin the immune system, which is the IB topic, I don't see how we can talk about the immune system and what your immune system is protecting you against unless we have some basic understanding of bacteria and viruses. So this video is going to focus on bacteria as a pathogen. Pathogens are anything that make you sick. So starting with the internal structure of a bacteria, what are some things we need to know about bacteria on the inside? We need to know that there is a difference between the types of chromosomes that bacteria have versus eukaryotic cells have. So there are two distinct differences, and you're going to be expected to know these two differences. The first being bacterial chromosomes are circular, whereas eukaryotic chromosomes are linear. Both, in terms of similarities, both are double-stranded. However, their shape is different. So bacteria have circular DNA, and you can see that right here. You can see that it's twisty, but it is a circle, whereas eukaryotic chromosomes, as we know, are linear and form, when they're condensed, form these chromosome shapes. But if you pull it out, you can see linear shape. The second thing that is different between a bacterial chromosome and a eukaryotic chromosome is the amount of protein associated with the DNA itself. So when you look at bacterial chromosomes, they only have a little tiny bit of protein, whereas our chromosomes have lots of protein. And so when we talk about what is that protein, if you go way back to DNA structure, and we talked about how DNA could wrap around histone proteins to form those structures called nucleosomes, that's where that protein is located. So you can see here we have our DNA wrapping around our histone proteins in order to condense the DNA into a smaller space. So you can see this blue line is the DNA wrapped around histone proteins. There's 8 of them in here, and this structure is called a nucleosome. And then these nucleosomes pack closer together so that you get this coiling, and that coiling ultimately condenses into the chromosomes. You've got DNA, you've got your chromatin, and then you've got all the way to your chromosome. So those are the two differences. One other thing that you will see noted in here is that bacteria, they do have extra DNA called plasmids, which we're going to be talking about. But in terms of chromosome structure, bacteria is circular, eukaryotic chromosomes are linear. Number 2, bacterial chromosomes only have a little protein, whereas eukaryotic chromosomes have lots of protein. Now, when you set up answers to IB questions, make sure that you keep things together. So you're saying bacteria have this, eukaryotes have this. Bacteria have A, eukaryotes have B, as opposed to bacteria AB and then eukaryotes, AB. I would literally say bacterial chromosomes are circular, whereas eukaryotic chromosomes are linear. I would always pair those things together. Let's talk about that plasmid. So those plasmids, not all bacteria have to have them. The number that they have can be different. It'd be 1, it'd be 2, it'd be 3. It's not like a set number, but these small little tiny extra pieces of DNA, this is not a chromosome. This is an extra circular piece of DNA. Could have a few genes on it. It can divide by itself, and we'll talk later about how one bacteria can pass this extra piece of DNA to another bacteria. Like, that is possible. So they're totally separate from the chromosome itself. So please don't mix those up. If you were able to do the Amgen lab where you had micropipetters and you were dealing with a plasmid, and you cut the plasmid, you ran it through a gel, you had the plasmid and you inserted it into a bacteria so that that bacteria made a protein that it had never made before, and you ended up with pink colonies growing on a plate. Like, awesome. I know not everybody has had the chance to do that, so we'll come back and revisit that later. But if you did do this lab, that was a plasmid. So how do bacteria divide? So in terms of when you talk about how do you make more copies of bacteria, how do you get more bacteria, they divide through a process called binary fission. This is an asexual process. This is identical copies. So you have one bacteria dividing and splitting into 2 bacteria, but these 2 bacteria are identical to the guy up here. Could there be random mutation so that there's a difference? Yes, that can happen. However, without mutation, this guy is identical to this guy and this guy. So if you start with 1 bacteria and it divides into 2, which divide into 4, which divide into 8, etc., divide, and you end up with like a mountain, like a little pile of identical bacteria, that is called a colony. So when you look at a bacterial plate here, where bacteria has been or a sample has been smeared across basically bacteria food, and then you wait 24 hours to see, maybe 48 tops, you wait to see how many bacteria are there. Was there bacteria on what you sampled? And yes, there was bacteria on what I sampled. And if you zoom in, you can see here's one colony, second colony, third colony. This colony started as one. So you can equate one colony to one bacteria. You now just have a pile of identical bacteria. Since you're splitting from 1 to 2 and having an identical copy, why can't we call this mitosis? So mitosis, remember, is a very strict definition. It's division of something. Mitosis is division of the nucleus. So mitosis is division of the nucleus, and we know that bacteria don't have a nucleus. So therefore, when you're talking about this process for bacteria, you must call it binary fission. You cannot call it mitosis. So if we're talking about bacteria just going through this asexual reproductive process, how do we get new combinations of bacteria? So we're going to talk about 3 different ways that prokaryotes can become genetically recombined or can become genetically different. The first way that we're going to talk about is called transformation. And in transformation, a bacteria is going to be taking in foreign DNA, as in DNA that wasn't part of itself, from the environment outside. So for example, this is our bacteria who is going to be transformed, and another bacteria broke down, died, fell apart. So now there's DNA bits floating around in the solution outside of it. And it is possible sometimes for a bacteria to take in that foreign DNA. And then if that DNA becomes incorporated into the chromosome, now this bacteria has extra DNA and potentially the possibility of making proteins that it didn't make before. So it could be where this foreign DNA becomes incorporated into the chromosome, or it could actually be where it just takes in a plasmid from the outside environment. So if it takes in a plasmid from the outside environment, it now has extra genes and can make proteins potentially that it didn't make before. This little experiment over here, this is Griffith's experiment that you learned once upon a forever day ago in honors biology, where he was studying a pneumococcus bacteria, like causing pneumonia and what can kill organisms. He was trying to figure stuff out. And so he had one strain of this pneumococcus bacteria. It was the S strain. And when he injected it into a mouse, it dies. It might be the R strain. I'm sorry. I'll have to go back and check that. I'm going to pause. Yes, this is the S strain. I was right. So the S strain is virulent, as in deadly. And so you can see when this bacteria is injected into the mouse, it dies. When you take this avirulent, not virulent, not deadly strain, it's the R strain, they inject it into the mouse, he lives. We know that if we take this deadly strain, heat it up, and destroy the bacteria, denature all those proteins, give that to the mouse, he lives, which makes sense. We know that we can, boil water, for example, kill the bacteria in the water so that make it safe to drink if there was some kind of an emergency. If you now take, though, this guy here who you heated up, killed all the bacteria, it's dead, won't kill the mouse, but mix it in with this group up here, this group right here that normally also would not kill the mouse, this guy becomes transformed into something that will kill the mouse. And so what happens is in this guy here, we have floating around pieces of DNA that then enter into your not dangerous strain of bacteria, just like this, and now he is able to produce those proteins that actually kill the mouse. Poor mouse. Second way that prokaryotes can take on new genetic information is through a process called transduction, and this involves a virus or a phage. So that's another name for virus. So these viruses are specific to bacteria. They're bacteriophages. They are viruses that infect bacteria, and you can see this little virus lands. It's like a little spaceship lunar lander, injects its genetic material into the bacteria. And when that happens, normally this bacteria would be told, you're going to make a whole bunch of new virus parts, and then those viruses will exit the cell with their new virus parts. Well, as part of the process, if this genetic material from the virus causes the breakdown of some of or breaks down the chromosome, for example, it's degrading that DNA, it could take a little piece of that DNA. When these little new virus parts are reassembled, it's going to grab virus DNA like it's supposed to, but it could grab a little chunk of bacteria DNA at the same time. And so now he's going to carry that bacterial DNA to another bacteria and incorporate that DNA into that new bacteria. So he's passing in this different section of DNA. And in this case, this bacteria here did not go through the whole process of having to make viruses. Something about maybe this incorporated DNA changed its structure a little bit, and this new bacterial DNA actually is now part of the chromosome. And so that is another possible way that a virus can play a role in passing genetic material from one bacteria to another by accident. And the last way that we're going to talk about is called conjugation, and it uses plasmids. So since plasmids have the ability to divide on their own, if there are two bacteria in the same vicinity, the same species, one of the bacteria has a way that it can send over kind of like a tunnel, and this tunnel is called a pilus. And so this little tunnel connects one bacteria to another bacteria, and a plasmid can self-replicate. So you can see it self-replicating, and it sends a plasmid over to the other one. This is a one-way pass. We're not going to do an exchange, so it's just one guy sending it to the other guy over here. This is as close to like bacterial sex as you can get, because there is like an actual connection from one bacteria to another. It obviously is not, but that is about as close as you can get. So I'm going to pause the video here, and I'm going to show you an animation. And it's going to be a little choppy because I'm not going to play the whole 5.5, 6 minutes. I'm just going to show you little chunks in case it's easier for you to visualize these processes as opposed to just hearing me describe them. Okay, so next one is going to be transduction.
Another form of horizontal gene transfer is called transduction. We speak about transduction when the transmission of genetic material between cells is carried out by viruses. In nature, this happens when a bacteriophage infects a bacterial cell. The viral nucleic acid is entering the host cell and is integrated in the bacterial DNA. Upon excision, errors may occur when parts of the bacterial DNA are additionally excised by accident. When virus assembly takes place, it may happen that bacterial host DNA is also encapsulated by accident. The phages are released from its host cell. This bacteriophage can infect the next cell, the recipient. The nucleic acid, this time also containing bacterial genes, again enters the cell. This sequence is again integrated into the recipient's genome. Genes from the host cell have been successfully transferred to the recipient cell by a viral vector. In the lab,
okay, and last but not least, conjugation.
Cell-to-cell contact of two bacteria. It is unidirectional. One bacterial cell is the donor, the other one the recipient. Besides the chromosomal DNA, the donor contains the fertility factor, a sequence found on the extrachromosomal F plasmid. The donor is considered F positive. The recipient, however, has no such F plasmid. This cell is F negative. F positive cells are able to form an extending structure reaching out to neighboring cells. This extension is referred to as the sex pilus. This protein tube attaches to the recipient cell and brings the cells together. One strand of the plasmid DNA passes via a channel from the donor to the recipient. After replication, the double-stranded DNA is present in both cells. In this case, the recipient also contains the F factor after transfer. From now on, it can also be considered F positive.
Okay, so hopefully seeing it was more helpful than just reading about it. So let's move to the external cell structure for bacteria. So bacteria have cell walls. Yes, plants have cell walls also, but the structure is different. The function is the same: maintain cell shape, provide protection, prevent bursting in a hypotonic environment. So we have the same function, but structurally there are differences between those types of cell wall, and fungus has a whole different kind of cell wall than either of these. The cell wall of bacteria contain a material called peptidoglycan. So it's not cellulose. That would be plant cell walls. Peptidoglycan is what is found within the cell structure of bacteria. When you look at that, there's more than one kind of cell wall. So it's not even just like plants have a cell wall. And yes, we know they sometimes have the secondary cell wall, and so you can go into that level of detail. But bacteria, there are two main kinds. You can see there's, whoa, tons of peptidoglycan versus, oh, a little tiny bit of peptidoglycan. And you can see here, there's all this cross-linking between these different strands of peptidoglycan. So how do you tell the difference? Obviously, if you're just looking at cells under a microscope, like how do you tell if the bacteria you're looking at, is it one that has a lot of peptidoglycan or one that does not have a lot of peptidoglycan? So you're going to go through a process called a Gram stain test. And this stain, it sticks to peptidoglycan. So there's a whole series. It's actually not that difficult, where you have on a microscope slide with the bacteria on it, and you add, like you heat it up, you do some rinsing, add some different series of stains, rinsing, blah, blah, blah. And at the end, dun, da, 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 you either have a darker color or a lighter color. Gram-positive bacteria are considered, you'll show as darker, and gram-negative will show as lighter. If the stain is sticking to peptidoglycan, gram-positive being darker, you guessed it. This guy has the gram-positive. He's gram-positive, lots of peptidoglycan, whereas gram-negative is that little teeny tiny bit of peptidoglycan. So that's ultimately, you're like, okay, so what's the big deal? So gram-positive have a lot of peptidoglycan. Gram-negative only have a little bit of peptidoglycan. But you can see structurally there's some differences here besides just the amount of material in that cell wall. Gram-negative bacteria, because of this structure, actually makes them more dangerous. It's harder for antibiotics to get across that cell wall. So if you let me go back to this guy here, because I think it's a little easier to see. The gram-positive bacteria doesn't have another cell membrane up here, so it's easier for antibiotics to get across. These guys over here, they have a cell membrane at the top. I know they have green up here, but there's phospholipids, peptidoglycan, more phospholipids. That is going to be harder for an antibiotic to get across. So it makes them a little more dangerous. So you can see if you were infected with something and they need to know, okay, what kind of bacteria are we dealing with? Then the first thing they can do that's really fairly quick and easy is, let's do a gram stain test. Are you in the gram-positive category or the gram-negative category? And it's going to start weeding you out. Okay, if you're gram-positive, then these are the options for you. If you're gram-negative, then these are the potential options for you. So since we're talking about antibiotics, easier to get across gram-positive, harder for gram-negative because of that cell wall structure, what does an antibiotic do? Like, how is it interfering, or why is it killing the bacteria? So if we're going to talk about one bacteria or one antibiotic, specifically penicillin, and his job, he competitively inhibits the enzyme that's making those cross-links for peptidoglycan. He's the enzyme that's allowing for the formation of the cell wall. So remember, competitive inhibition is when you get in the way of the active site, so of the material itself. So if you can't make the end product because you have a medicine that's now in the way, and so notice there's the normal substrate, and then here's the drug, the antibiotic. He's very similar. He's going to bind up or he can either sit in the active site or bind up part of the substrate. So it can work in more than one way. But in competitive inhibition, remember, you just sit into, dun, da, 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 I'm in the active site, therefore you can't make the end product, you can't make that cell wall, and as a consequence, the cell wall is weaker and the bacteria dies. So how do bacteria make us sick? There are two different ways that bacteria can make us sick. One, like tuberculosis, actually gets into your tissues and causes the destruction of those tissues. Most, however, bacteria are making us sick because of the proteins and things that they produce. So they're producing some kind of toxin, and it's the toxin that's actually making us sick. So the bacteria is just living its little bacteria life and, producing material through exocytosis, and then those toxins are what are causing our cells to have damage. So for example, cholera how cholera makes people sick is that you have ingest the cholera bacteria. The bacteria produces the toxin. So now we're in the intestine. So you're in the tubes of your digestive tract. And this toxin then binds to a receptor of the cell that lines your intestine. So this is the space like where your food and water and stuff would be. This is the cell making up the intestine, and it causes a whole bunch of stuff to happen. This is called signal transduction. We'll come back to that later. So it causes a whole bunch of stuff to happen, but at the end of the day, notice we are sending ions out into the space. Well, you know that if you send ions out into a space, water is going to follow, and because water follows, you end up with a lot of water in your intestines, and this is going to cause diarrhea and massive dehydration. So cholera is just causing huge amounts of dehydration, and to the level where if you have enough cholera toxin, you actually can die from cholera, especially if you don't have access to the healthcare that would help you get better. But ending with that, I don't want you to, as we end bacteria, I don't want you to be like, oh, all bacteria are bad, they're all making us sick, we're taking antibiotics, or what are we going to do? We use bacteria in our everyday life. So besides natural uses, I'm talking about not the natural stuff, because when we get into digestion, we will talk about how there's bacteria helping us, live our life, and bacteria are a very important part of our digestive system. I'm talking about the application of bacteria, like in society, not within you. So we use bacteria for research. So how did we learn about, DNA replication and protein synthesis? We didn't use a eukaryotic cell for that. We used bacterial cells, prokaryotic cells for that. So that's how we learned about how some of these metabolic functions. We do a lot of research on different proteins and different genes through bacteria. When you look at bioremediation, it is possible to use bacteria to help break down bad things in our environment. Is it going to be a fast process? No, it is going to be a very slow process. This isn't like, oh, too bad, there's a big oil spill, we'll just throw some bacteria in the ocean. You're going to mop up as much oil as you can. However, if you can throw in some bacteria that can also break down that petroleum product, it will help over the long period of time. So it's more of like the extra help as opposed to like the main way. Another way that we use bacteria in society is to help get more metal out of ore. So you're going to dig up a whole, we don't need to know all this process. This is just in case you're curious. You just dig out a whole, tear down this Rocky Mountain stuff and send it through a whole big process. You're trying to get the metal out of that rock. Bacteria can help get more of the metal out. We also use bacteria to help us make antibiotics. That's kind of ironic, here using bacteria to make a product that's going to kill other bacteria, but we use bacteria to help make antibiotics, to make vitamins, and even to make some hormones. So when you're looking at just medicines and things that we use, there's a definite pharmaceutical component. And then we use bacteria just in terms of food making. So if you're on yogurt, if you look at the side, it'll be like live and active cultures. In other words, there's a lactobacillus bacteria that's in there. Good for your digestive tract. Yay, go yogurt. But we use it to help make the yogurt because it's going to give it that fermented, tangy flavor. Cheese, we use bacteria to help us make cheese, like go cheese. So we use it in, and we've been using bacteria in food production for a really long time. These other ways and these ways are definitely much more modern. So we're going to pause here and pick up on viruses at another time. Have a good day.